रोहित जी, जो हमारे स्पीकर हैं, भारद्वाज साहब ने ज्वाइन कर लिया है। आप उन्हें को होस्ट बनाइए। ठीक है। आप BHARA करके सर्च करेंगे, आ जाएगा। ठीक है।
अच्छा।
जय श्री राम सर, स्वागत है आपका। भारद्वाज सर, जय श्री राम, सुन पा रहे हैं आप हमें? सर नमस्कार, भारद्वाज सर, सुन पा रहे हैं आप हमें?
हेलो सर, नमस्कार। मैं डॉक्टर नीरज कुमार बोल रहा हूँ, IRI से।
अच्छा। जी जी जी।
डॉक्टर भारद्वाज सर मीटिंग में व्यस्त हैं।
सबसे पहले तो आपको बहुत बहुत बधाई।
थैंक यू सर, थैंक यू सो मच। एक्चुअली
जी
उन्होंने
जी।
मुझे बोला है कि आप प्रेजेंट कर देना। वो कोई मीटिंग है हमारी हॉस्टल्स की,
अच्छा।
डायरेक्टर, तो
अच्छा।
वो वहां से उनको
ठीक है,
समय।
कोई कोई बात नहीं डॉक्टर साहब, आप कर देना, आप कर देना।
हाँ, तो उनकी ही स्लाइड्स है, वो मैं बोल
ठीक।
दूंगा।
ठीक है, ठीक है, ठीक है। आज आप अपना अचीवमेंट भी बता देना, 2 मिनट, फिर शुरू करेंगे।
अरे सर, क्या है।
जी
ठीक है।
जी जी। ठीक है, ओके।
थैंक यू सर। प्रेजेंट करना है अभी कि थोड़ा टाइम है?
बस सर, 2 मिनट बाद मैं आपको खुद बुला लूंगा सर।
ठीक है, ओके सर, ओके।
ठीक। नीरज सर, मैंने अपना व्हाट्सएप नंबर भेजा है, जरा इस पे व्हाट्सएप पे हेलो का मैसेज भेज देना। नीरज सर, आप सुन पा रहे हैं हमें?
जी सर, भेजता हूँ जस्ट।
हाँ प्लीज, हाँ हाँ हाँ। नंदिनी, मेरी आवाज आ रही है?
हाँ जी सर।
ठीक है, आप स्टार्ट कर सकते हैं।
Okay, sir. A very lovely good evening to all our respected dignitaries, distinguished speakers, mentors, and participants joining us from India and across the globe. I'm Nandini Sharma, Assistant Director HR at AgriMeet Foundation Bharat. Warmly welcome you all to the day 11 of International Winter School Bharat 2025. As we continue this global journey of learning and collaboration, today's session spotlights innovation driven crop improvement with a focus on pulses, combining classical breeding with modern genomics to advance food and nutritional security. So, about the winter school, it is jointly organized by AgriMeet Foundation Bharat and UGC MMTTC Jodhpur in collaboration with leading ICR institutions in universities and international research partners. With the theme Boosting Holistic Agriculture Through Research, Awareness, and Training, this initiative provides a platform to explore cutting edge advancements in plant breeding, biotechnology, precision agriculture, and sustainable food systems. So it is an honor to introduce Dr. Neeraj Kumar Tiwari ji, chickpea breeder at ICR, Indian Agriculture Research Institute, Pusa, New Delhi, currently contributing to Institute's flagship chickpea improvement program. Dr. Tiwari joined the Agriculture Research Service in 2017 and has since focused on genetic enhancement, trait integration, and product development in chickpea, spanning Desi and Kabuli market classes. He has been associated with the development of 4 chickpea varieties, 3 Desi and 1 Kabuli, aligned to agroecological needs and market preferences, with emphasis on yield stress tolerance and disease resistance. His scholarly output includes around 25 research papers in reputed journals reflecting impactful work across breeding physiology and applied genomics in chickpea. Dr. Tiwari's academic excellence has been recognized with the IARI Merit Medal for PhD, underscoring his strong foundational research training and scientific rigor. Most recently, he has been honored with NAS Young Scientist Award 2026 in Crop Sciences, a prestigious national recognition for early career scientists demonstrating outstanding promise and contributions. His work complements IRI national significant efforts in chickpea, including drought hardy, genomics assisted varietal developments for central India, strengthening resilience and productivity under moisture and stress. Through sustained breeding and collaborative testing, such as pipeline support, farmer centric impact, and bolster India's leadership in pearls improvement. We are truly delighted to have you with us today, sir, on the behalf of AgriMeet Foundation Bharat and UGC MMTTC Jodhpur. We extend a warm welcome to Dr. Neeraj Kumar Tiwari ji to the International Winter School Bharat 2025. We look forward to your insights on chickpea breeding, strategies, trade, pyramiding, and the path from discovery to deployment in India's pearls research ecosystem. He is speaking on the behalf of the other speaker, Dr. Bharadwaj sir. So, ladies and gentlemen, please join me in welcoming Dr. Neeraj Kumar Tiwari ji. The session is yours.
Thank you. Thank you so much for a very nice introduction.
सबसे पहले बहुत बहुत मेरी तरफ से भी बधाई। ये पूरे IRI के लिए गर्व का विषय है कि आपको नास का जो है यंग साइंटिस्ट चुना गया।
बहुत बहुत धन्यवाद सर, बहुत बहुत धन्यवाद। Basically, I will be speaking on behalf of my Guruji, Dr. Bharadwaj ji. Basically, he is very busy as he has been bestowed with extra duties. Like he is now MOHR. We call it as, in other university we call it as director of student welfare. So
Yes, yes.
he is now acting as MOHR in IRI, and the whole of IRI students, their accommodation, anything related to the students, he is responsible person in IRI. So he's very busy. I'm sorry for that. And he just informed me that you have to go and deliver on behalf of me. So I am here. Just a disclaimer that I will not be able to do a justice whatever he has prepared. I am very new to this area, just 7 or 8 years of experience, but he has more than 25 years of breeding experience. So I will be trying to do my best. Okay, so can I share the slides?
Yeah, please, Dr.
Okay. Just a second. Okay. So is it visible?
Yes, visible, Dr.
Okay. So basically, we at IRI in chickpea breeding program, we focus on development of chickpea varieties that are suitable for farmers across the India, as well as we are also focusing on the consumer need. So varietal development itself is a complicated task, as in conventional breeding approaches, varietal development used to take around 8 to 10 years of breeding efforts. After that, the variety goes into a trial system. And in India, we have all India coordinated research projects for different crops. Likewise, we have impulses. So the identified material, the best selected by breeder, goes into that all India coordinated research program. So there, the independent trial again is conducted for at least 3 years, and there are different systems like IVT. First, you have to enter your entries to IVT, that is initial varietal trial. Then, if the entry performs certain best, that limit is fixed, that is 5 to 10% more than best checks available. If the variety surpasses that, it is again promoted to advanced varietal trial 1, and then again a check is included, and if the variety again passes through check, then it goes to advanced varietal trial 2. Then among in advanced varietal 2 also it is evaluated for its performance, and that is how a variety is released. So for an average breeder like me who joins, it takes around 13 years to breed a variety. And then after release of the variety, again it takes 3 to 4 years to go to the consumer, that our stakeholders, our primary stakeholder are farmers, who are our, in India, we target farmer, so they are our primary stakeholders, and they take us our varieties and they grow them. So the variety reaches them till 14 or 15 years. So this is a very vast length of breeding, and basically it is being utilized as commercial breeding everywhere in different crops. So here at IRI, we try to include other breeding techniques or other breeding option, that is genomic assisted breeding, that we try to improve the best of the best varieties available for some distinct traits. That is a simple back crossing, and we include the molecular markers in that. Then it becomes molecular assisted back cross breeding, and in that technique we are able to release the varieties in 6 or 7 years. So we are cutting the time by 50%. We are reducing the breeding length by 50%, and we are able to give our varieties very early to the farmers. So this is what we are doing at IRI, and our Dr. Bharadwaj is leader in that, and he is our program leader, and he has developed more than 28 varieties in chickpea. And but we have developed around 8 varieties through this advanced genomic assisted breeding tools. So I will be speaking on the topic, the genes to varieties, the chickpea genomic assisted breeding, a saga of success. The whole presentation is prepared by him. I'm just speaking on behalf of him. Okay. So basically, we all know as an agriculture scientist or agriculture worker that there is challenge in our agriculture system, that we have limited resources and more mouths to be fed. And India is one of the highest populated country, and the population growth rate is also very high in our country. That is 1.74 percentage. And we require more food, but our resources are shrinking. We have less water, less arable land, and less labor. So these are the challenges we have currently with us. So keeping in the mind, we have to deliver and we have to feed our population. So these are some of the major challenges. And so what we look at in breeding program. So in breeding, basically, in conventional breeding, what we do, that is a whole pipeline of breeding is there. So we look at genetic diversity, and how we evaluate genetic diversity, that is visual appearance of the genotypes. And we screen accessions for the trait of concern and the trait which we are interested in. We try to discover or we evaluate the germplasm and we classify them according to their traits. We also, there is some biometrics involved in that. We try to evaluate the performance of the individuals. That is, we try to estimate the breeding values and the best breeding values, which is estimated by progeny performance. The individual are selected in crossing program, and they are now brought to crossing program, crossing block. That is basically a dream box of a breeder, the crossing block, and we try to mate them. And after that, there is whole sort of chain that variety that the genotypes we cross them. It comes to F1, F2, F3, F4, F5, F6. Then a fixed line is evaluated in replicated trials again, and the selection is made on every steps. Like from F1, we try to identify the hybrids, and in F2, we try to select the best individual. Then F3 again selection is performed, F4 again selection is performed. So selection is pressurized at every stage. And with all these techniques, we are able to improve some of the genotype for our trait of concern. And then the improved genotype goes to seed system and then to market to the consumer. So this is the whole pipeline that is followed in breeding of a new varieties. Or just a second. Okay, so this is how the translation of genomics in agriculture takes place. We have a repository of different accessions at our gene banks. So in gene banks also, we sometimes we make some exploration trips to different and we try to identify accessions that are useful to us. And mostly the accessions which we require, we directly use the gene bank collections. And you will be surprised to know that more than 95% of gene bank collections are underutilized, or they are not utilized fully by the breeders, because we house large number of collections, but we are not able to characterize them. And we, after characterization, so we take very limited amount of accession that are used in breeding program. So to see the whole length of diversity in germplasm, we try to make core, mini core, and trait specific collections. And also we try to develop some genomic resources that acts as a reservoir for our breeding program. So in genomic resources, we have different types of recombinant inbred lines, integration lines, multi-parent advanced generation intercross lines that we utilize as a breeding program. From all these germplasm resources, we try to mine the actual cause of that trait. That is by identifying some QTL regions, that is quantitative trait loci, that is basically linked to the trait of concern. And we identify the closest markers that are linked to that QTL regions. We also try to mine marker trait association, that linked marker to the trait, and different other techniques like genomic estimated breeding values, quantitative trait, nucleotides. All these are identified, and these acts as a genomic resources which we are able to utilize in our genomic assisted breeding program. And in genomic assisted breeding program, it's basically same as conventional breeding program, but the difference is that we are utilizing some of the linked markers or linked markers to that trait, which we have characterized, and we are directly selecting for those markers here. So that comes in the genomic assisted breeding. So we have mass, that is marker selection. The selection is just supplemented with marker. So our selection becomes more precise as compared to the conventional selection or just visual selections. So we screen the genotypes for the individual that carries that particular marker selection. Then if we want to incorporate more number of you can say some for quantitative traits, we have the markers which have very limited number of phenotypic variants explained, or the QTLs which are having very limited number of phenotypic variants. So in that case, we try to accumulate those markers in a single genotype. So in that case, we use marker restricted recurrent selection. And the advanced version of marker restricted selection is your genomic selections. So in genomic selection, we try to identify the genomic estimated breeding value on the basis of the marker distributed across the genome of individual. And the best individual which we identify on the basis of genomic estimated breeding value is being used in our breeding program. So this is how the whole process, and in each step we have an analytical and decision support system. Also nowadays AI is being also utilized, and some of the experiments are being done, which model is best, what is happening. So all these things go in hand to develop a variety. So here is our pre-breeding approach. How do we come from germplasm to a variety? So in pre-breeding approach, we collect, evaluate, transfer, and deploy. We collect some new accessions or some novel genotypes are collected. They are evaluated for carrying some good genes or valuable QTLs, and then it is transferred into breeding program, and then it is utilized in our breeding trials or in breeding pipelines. So basically, we are at chickpea breeding program, we target for biotic and abiotic stress. So abiotic stress basically includes heat, drought, frost, cold, and salinity. These are our major abiotic trait of concern. And in biotic, there are different diseases in chickpea. Like we have most important disease, that is wilt, Fusarium wilt. And after that, we have Botrytis gray mold, Ascochyta blight, dry root rot. These are some of the important biotic diseases that affects chickpea cultivation. After these, we have some novel traits that we are looking at, like machine harvestability. As I told earlier, there is a very much labor problem that in coming days we are not going to get agricultural laborers for our work. So we are developing some varieties that are amenable for mechanical harvesting. So in this is one of the unique trait that we are focusing. Then also we try to fit our chickpea in different cropping systems. So in that case, we need extra short duration varieties which have very short duration, like 90 to 100 days, that can fit into different cropping system like rice, fallows, and all. And also we are looking at different plant architectural traits like high harvest index, lodging resistance, then determinate type. So these kind of trait we are focusing in our breeding program, along with the traits which concern our end user, like its nutritional value. So we are also looking on some nutritional quality traits like protein content, mineral content, all these traits we are looking here. So as a chickpea, let me introduce the chickpea. You all might be aware of this chickpea. So chickpea is one of the important grain legumes as far as Indian growing condition is concerned. It is one of the important pulse crop, contributing more than 50% of total pulse volume. And among the pulses, it is also known as king of pulses. And because of its high nutritional value, it has very high protein, about 24 to 25% we got in general chickpea. It has high fiber, low sodium, and the protein has unique ability to act as a functional food in protein based market. So the protein of chickpea has very balanced amino acid profile, so that make it more relevant. And apart from that, the various medicinal values of the chickpea make it more important, like it promote heart health, improve digestions, control blood sugar levels. These are the important aspects of chickpea. Currently in India, chickpea is cultivated about 11 million hectare areas with the production of 13 million tons. So this is the one statistics of chickpea. The drought, as we discussed earlier, the major abiotic stress in chickpea, it can cause up to 50% of yield losses. As we know, the chickpea is one of the most important rain-fed crop in Rabi season. The areas where no other crops can be grown, the chickpea is preferred, as it is mostly 50% of chickpea area is sown under residual moisture conditions. So there is no availability of irrigation, or very limited irrigation is available. So under those conditions, chickpea is planted. And the most important drawback of that regions is that those regions are drought prone. So drought is one of the most important constraint that affects its production. So we here try to do customized breeding or breeding program to enhance drought adaptation. Like we focused on root architectural traits, the genotypes having deeper going root with spreading type, we prefer those, and we have phenotype some of individuals from our germplasm, and we have identified the genotypes that have good root traits. That along with that, we are also trying to focus on physiological parameters that will act as a secondary traits that can be used in as a donor in breeding for drought resistance in chickpea. Those includes membrane stability index, also relative water content, along with the stomatal density. All these traits we take into consideration while identifying for a robust donor for drought tolerance. So basically, we also here look at land races and wild relatives, trying to mine different novel wild relatives carrying unique traits and unique alleles for the trait. So currently we have 7 wild relative species, include Judaicium, Pinnatifidum, Bijugum, Reticulatum, Croatium, Cunetum, Yamashite, along with 162 land races. And we try to classify these wild relatives in trait-specific collections. And for different traits like drought, we have different collection. For nutrition and protein quality traits, we have different wild relatives that can act as a donor. So basically, they have we are trying to identify allelic variant of the gene or the trait in our wild relatives and germplasm collections using these approaches. And after analysis, we have from the global collections of chickpea, we have classified our chickpea material into different clusters. These are our in-house collections that we are having, and we are utilizing this in breeding program. One of the earliest variety, that is USA 1103. That is one of the first varieties that had been developed by utilizing a wild species, that is Cicer reticulatum, was utilized. And this is highly drought tolerant genotype that was earlier developed using conventional breeding tools. And it is one of the best variety that is utilized as a parents in our recent breeding programs. So as I told earlier, we are trying to look at the allelic variants of the gene in these land races and in these wide relative cultivars. So for that, we try to mine the allelic variant in different gene background. That is, here we are reporting some dihydrin gene, and we try to see the sequence variation of this dihydrin gene in our different land races and wild relatives. And we could find some of the tolerant individuals carrying some unique SNP that can be used to develop a SNP-based marker, and it is being utilized in our breeding program. So this is the example of another gene, that is ASR gene. That here also in ASR gene also we are trying to mine allelic variant, and that is being utilized in our breeding program. So these are ASR gene Ramachandran plots and its protein folding parameters. So by using all these genes, we have identified one of the most important donor, that is ICC 4958, that is having let's say well considered donor for drought tolerance, having unique trait like root adaptation traits. Along with the trait, the IC 4958 QTL hotspot also harbor trait for high yield, high harvest index, and high seed size. So most of the breeding program which we use for developing drought resistant individuals, so the donor is IC 4958 because it has very limited linkage drag, as it is one of the well adapted genotype, but also having the unique traits of drought tolerance. So the Varshney et al. Have fine mapped this QTL hotspot, and this QTL hotspot have been flanked by different markers which are being routinely used in breeding program. So this is the QTL fine mapping of this QTL hotspot region that contains more than 13 genes in the flank marker between the flank markers sequence. And these genes not only contribute towards the drought tolerance, but also towards the higher yield. Like they have genes responsible for high harvest index and seed size. So that also contribute to the yield enhancements in chickpea. So this work was done earlier by Varshney et al. Along with this, they have also classified 3,000 chickpea accessions, and they have tried to mine these QTL hotspot on the basis of QTL-seq analysis. And this is how our breeding pipeline looks like. We first make the cross between elite and donor, and F1 we grow at off season. So in 1 year we are trying to take 2 seasons. So in 4 year we are completing all BC2 and F1. In 5th year we make BC3 F1, and 6th year BC3 F2, and in off season BC3 F3. So in we are able to complete all breeding cycles in 4 years, and the trial takes another 2 years. So within 6 years we try to come up with a new variety using this marker-assisted backcross breeding approach. So this is example of Pusa 10216, which is improved for drought tolerance, which was high yielding variety earlier developed by IRI, that is Pusa 372. And Pusa 372 was introgress by QTL hotspot using donor IC 4958, and we developed this Pusa 10216. This the variety Pusa 10216 is one of the first molecular assisted bred variety in chickpea and pulses, which was developed by Dr. Bharadwaj, who is our lead at IRI. And also we try to evaluate our material by using different physiological techniques so that we are sure for its characters. So first we try to evaluate genotype at a rainout shelter, at glass house, at fields, and at LISI scan platforms to see what are the different traits that respond under drought, and we then identify the good donors that have a unique trait along with their physiological performance, and then we use in breeding program. So this work was done by Dr. Bharadwaj and its collaborator Dr. Vashni Rutvik, where they try to find map this QTL hotspot, and they identified a novel hotspot region that is TEFI 2.0 that is responsible for this drought tolerance in chickpea. So this is again the example of or the performance of PUSA BGM 10216 under All India coordinated trials, and the variety has unique potential and it has performed superior to its recurrent parent. More than that, in moisture stress conditions, the red lines which you see is percentage of moisture at flowering and moisture at maturity. Green line depicts that. So the introgress line, that is BGM 10216, is superior as compared to PUSA 372. So PUSA BGM 10216, also known as PUSA 1026, was the first MABC lines released for India for commercial cultivation in Central Zone, and it is the near isogenic line or integration line of PUSA 372 that was developed by collaboration with ICRISAT and has 16% yield advantage over recurrent parent across the test center. Also, it has high seed weight, that is 24 grams. As I earlier told, that the QTL hotspot also carries gene for seed size along with the drought tolerance. And also this variety is having good protein content, that is more than 22.6% protein content, and is one of the highest indented variety from the rain-fed zone of country. So this variety was very popular and has got wide publicity, and it was one of the first product that was developed by IRI, ICR and ICRISAT collaboration and was quite popular. Along with that, another line having the recurrent parent of DCP 92-3, that is known as IPC L4-14, that was developed for Northwest Plain Zone by the same mechanism as I described. And along with that, one extra line, that is PUSA 4005, that also for Northwest Plain Zone was developed using the same donor, that is IC 4958, and the same linked markers were utilized in breeding these two varieties. Another variety that is very recent, that is PUSA JG16, which is one of the recent product, and its seed indent is more than 100 quintals, and this variety was the improved version of JG16. JG16 is a popular variety developed by JNKVB Jabalpur, and it used to be one of the mega variety in that region, but became susceptible due to sporadic drought, and farmers didn't like its behavior under drought conditions. So we try to improve this variety as well for drought tolerance. And recently we came up with PUSA JG16, which is having high drought tolerance. And along with that, the short duration is also incorporated, and the variety is having 110 days of maturity seed to seed. And under drought condition, the variety is performing very well, more than 2 tons per hectare or 20 quintals per hectare its yield under drought conditions. So the variety JG16 was also given wide publicity, and Dr. Bill Gates also was keen to see the drought tolerant materials that were developed by IRI. And as he visited the rain-fed areas of Latur, Maharashtra, and central India, he saw that the green chickpea that were flourishing without any irrigation. So he was interested to see the varieties, and he came to know that all those varieties are basically Pusa 10216 or Pusa GG16s that were developed by IRI. So he was very interested to visit IRI. Likewise, that Pusa 10216 or the IC 4958 as a donor was utilized in breeding different varieties across the globe. Like in Kenya, it was released in the name of Giletu. The same QTL hotspot was utilized in releasing that variety. And that simple QTL region or the QTL hotspot have bred more than 8 to 10 varieties globally, and it has contributed to enhance the yield in rain-fed areas. So this is the impact of molecular breeding, that it has no boundaries. Basically, due to efforts of ICRISAT, the QTL hotspot were transferred into multiple backgrounds in different countries growing chickpeas. So also in our breeding program, we try to make some product profile. We have some target production environments for regions, like we have major regions in central in South India, in Northeast Plains, and then Northwest Plains. These are our major target areas. And the priority traits for the central India is basically short duration. Because in central India, the crop matures quite early as compared to North India, because the winter is getting winter is early, and the summer is also early in central India. The summer onset after Feb or Jan end. So we need varieties that are highly resistant to terminal heat, along with their short duration nature, along with drought tolerance. And in biotic stress, we are looking at wilt is our compulsory trait. We try to maintain the wilt resistant in every variety. Along with that, if the variety is going to rain-fed area, then dry root rot. Basically, we don't have any resource for dry root rot resistance, but we are trying to maintain the moderate resistance in some genotype we occur. So we try to maintain the moderate dry root resistance in our breeding program, breeding materials. Along with that, the extra large seeded Kabuli is also preferred in central and southern India. And the new trait like mechanical harvesting and nutritional quality is also being incorporated. So these are some product profiles of that zone. Like in northeast zones, the late zone is practice, because after paddy we have a lot of areas that goes as a fallow. So if there is sowing of some crop, then the crop is very late. So late zone variety we prefer with short duration, along with the BGM resistance and high temperature and drought resistance. In Northwest Plains zone, cold is one of the major problems. So cold tolerance along with the Ascochyta blight and wilt is our main focus traits. So this is also the mapping of another QTL that is linked with Fusarium wilt resistance. So this is another study where we try to integrate the Fusarium wilt resistance using molecular assisted breeding tools, and we try to pyramid more than 3 genes using marker assisted breeding. This is the differential gene expression of wilt resistance in susceptible and resistant genotypes, and it is clearly indicating that there are different genes that play a critical role for imparting wilt tolerance in chickpea. So like that, I explained about drought. The similar scheme is practiced in for Fusarium wilt, and we have come out. You can see the wilt-seek plot. Basically, whatever we do, even we do marker-assisted breeding or anything else, we need a phenotypic proof. The genotypes which we select should be phenotypically superior than the best check at even if we tag it with molecular markers or we select using molecular marker, we again go for the phenotyping profiling. So this is our wild-seq plot, which acts as a major phenotypic screening facilities for wild tolerance, and we evaluate our introgress material here, and the material which start, you can see here we have PUSA 372, which have very sparse plant stand under wilt stress condition, but this is our integration line of PUSA 372 that's having very good plant stand under wilt stress condition. So these are our different integration line that had been developed in background with different genetic background with donor to wilt stress tolerance. So we have WR315, one of the major donor for wilt resistance. That WR315 have QTL regions that it carries multiple genes and multiple genes for wilt resistance, and it is used one of the donor in different wilt resistance program. So under this program, we have developed one variety that is known as PUSA chickpea Manav, that is BGM 20211, with enhanced fusarium wilt resistance. We have pyramided multiple QTL here, multiple genes, more than 3 genes were pyramided. And the major effect of pyramiding is that PUSA 391 is very high yielding variety, but has become susceptible to the wilt. So we try to pyramid it with WR315, and we have bred this variety that is known as PUSA Manav. It is one of the highest yielding variety currently in India. It is released for central India, and the potential yield is around 40 quintals per hectare, but average yield at farmers field is also more than 3 tons or 30 quintals per hectare. So this variety is having the highest seed intent as of now. This year we have got more than 320 quintals of breeder seed intent, and it's very popular variety among the farmers, and farmers from central India and from Maharashtra, from southern Rajasthan, they prefer this variety, and the seed demand of this variety is very high, and this is one of the product of marker-assisted breeding for biotic stress. So basically, marker for complex trait in plants, how to use the markers for the simpler traits that are easy to map, that are very that are governed by very few genes, that can be easily in progress. But for the complex trait like nutritional traits or traits affecting plant architecture, they have they don't have any fixed number of genes. They have a large number of genes, and some genes are having very minor effect against the phenotype. So how to use the marker in the traits that are affected by large number of genes? So we try to we have recently utilized the genomic selection methodology in chickpea. So before that, I would just like to give you a brief about the breeder's equation that you all might be aware of. That breeder equation, the breeder deals with a genetic gain. The parameter in which breeder is evaluated is genetic gain, and the genetic gain is directly proportional to selection intensity and selection accuracy and genetic variance. Genetic variance is basically the feature of germplasm, how your germplasm is handled, or how germplasm carries the gene or not. But the genetic selection intensity and selection accuracy are the most complicated thing that needs to be standardized in breeding, so that we can get maximum genetic gain. Along with that, time is also very important. So genetic gain is inversely proportional to time. So if we are able to take multiple generations in a single season or single year, then we are hastening our genetic gains and we are reaching to our goal very rapidly. So these are some of the important parameters that needs to be taken care while going for breeding. And in genomic selection, we are basically targeting selection intensity and selection accuracy. So these are earlier varietal release system where we have limited number of genotypes. Then we make F1, F2, F3, F4. If we have 2 seasons in a generation, then we require 6 seasons to get to station trial 1. And then again a year of testing. So at least a 10-year is required to breed a release a variety. This paper was published by Dr. A.K. Singh et al. And nowadays we are taking large number of genotypes. So our funnel is broadened. We are taking number of crosses, and we try to select the parents on the basis of their genomic estimated breeding value, and then we shorten our funnel directly here itself, and we select the individuals that are having higher genetic values, and we make the crosses good into good, and then we score them for genome-wide associated or distributed markers, and then we select the individual. So we are basically making it half of the time as compared to the earlier one. And if we incorporate speed breeding here, so if we can take 3 generations in a year using speed breeding facility, then this methodology will further reduce the time of breeding. So genomic selection based breeding methodologies are being implemented for the complex traits that are not governed by a few genes. So these are some nuts and bolts of genomic selection. So genomic selection, we have one training population, and another we have a breeding population. So we train our model. The marker is trained for the phenotype with phenotype using different models, and the genomic estimated breeding values of those marker is calculated on, and different models are used for that, statistical models. And in breeding population, we directly do the selection without phenotyping. We just use the markers which were modeled, and we try to estimate the genetic worth of breeding population without phenotyping at earlier generations. And we phenotype after selection or at later generation when we have very limited number of entries that we select. So this approach is now being utilized. So the optimizing the genomic prediction based selection strategies in chickpea, we are restructuring training population. We are genotyping. We have trait specific training populations for different traits. Like for drought, we have different training population, and for nutritional trait, we have different training populations, so that the prediction accuracy of the breeding population is enhanced. So if we utilize trait wise training population, then the prediction accuracy of those marker is enhanced, and our selections become more precise and more robust. Though genomic selection is not applicable everywhere, so it has to be utilized for the traits. Those are complex and those are hard to phenotype. Like for nutritional traits, if we need to phenotype them for nutrition, or we have to offer different nutrition, then it's a costly affair. Also, if we the genotyping, if we just do genotyping, then the large number of population we can easily eliminate, and we can select few individuals that can be a phenotype at later stage of their breeding program. So this is one of the experiment or the on the GS base was done by Dr. Bharadwaj et al. And they found that there are different models in genomic selection prediction, and they found that random forest model predicts best for the trait like drought susceptibility and index and 100 seed weight. The prediction accuracy was varied from 0.8 to 0.9%. That is very high. Recently, we are trying to predict the prediction accuracy of nutritional trait, that is coming up to 0.6 or 0.7. That is also considered as a good prediction accuracy. And breeding lines based on these breeding values were selected and the parents were selected on the basis of this genomic estimated breeding value to breed further. So this is the example of that breeding prediction accuracy of genomic assisted breeding value for different traits. And also there is one complex, the Desi training set predicts Desi breeding population good as compared to mixed genotypes that having Desi and Kabuli mix, and Kabuli predicts Kabuli good. So this is the different prediction based accuracy for the different traits. So we are trying to deploy genomic selection in our chickpea breeding program based on our results and pilot experiments from AICRP. We have initiated GS in our breeding program for the traits like yield, for plant architectural traits, for nutritional traits. We have made different training population, and that training population is being utilized to predict genomic estimated breeding values of different markers. And now these markers will be utilized in estimating the real worth of the individual, so that we can take the parental line on the basis of genomic selection in our crossing blocks and breeding proponent. So in brief, the next generation genomic approaches are very useful in developing climate ready varieties. And for a given crop, there need to be developed genomic resources, genetic maps, genomic sequences, assembly, so that it can be more precise and more perfect in breeding program. And germplasm collection breeding population can be phenotype for the trait of interest related to climate change, can be genotype with the high throughput genotyping technologies. And GWAS and link markers are utilized to generate the genomic resources or the gene that are linked to the trait. And genomic breeding techniques that are mass or marker selection, markers by cross breeding, and markers of recurrent selection, genomic selection, all these can be used to develop a high yielding chickpea varieties. So I am very much thankful to my team leader, Dr. Bharadwaj, who has given me opportunity to present his work on his behalf. Actually, he was very busy today. So on his behalf, I was given a responsibility to present. I may not have done justice to that, but I have tried my best. I am thankful to all the chickpea staff who are the backbone of our research program. I also want to thank our director, IRI, our head of the division, and all the students and research fellows who are involved in the chickpea breeding program, our collaborators and our funders. Thank you everyone for patience listening. Thank you.
बहुत बहुत धन्यवाद तिवारी साहब कि आपने सर के आह्वान पर, सर की अनुपस्थिति होने के बाद आपने जो है एकदम अनायास किसी को कोई चीज असाइन करी जाए तो अपने आप में वो एक चुनौती तो होती है, लेकिन सर ने फिर हमें मैसेज भी किया कि मैं फ्री हो गया हूं। आज उनका जन्मदिन भी है, तो मैं उनको जन्मदिन की बहुत बहुत शुभकामनाएं प्रेषित करना चाहता हूं, और जो हमारे पार्टिसिपेंट्स हैं वो भी उन्हें जन्मदिन की शुभकामनाएं देंगे।
जी सर आ गए हैं।
बहुत ही हम्बल, बहुत ही पोलाइट पर्सनालिटी है, बहुत ही हम्बल। इवन कभी भी हमने कोई भी सहयोग की बात सर से कही होगी, हमने मैडम से कही होगी, तो कभी भी किसी ने हमें डिनाई नहीं किया, हमेशा सर ने ऑलवेज सहयोग किया है। तो सर को तो जन्मदिन की शुभकामनाएं, और आपको नास यंग साइंटिस्ट की बहुत बहुत शुभकामनाएं।
बहुत बहुत धन्यवाद सर। बहुत
हमारे जो पार्टिसिपेंट्स हैं इस पर्टिकुलर टॉपिक पर अगर उन्हें कोई डाउट है, कोई क्वेरी है, तो इस समय डॉक्टर भारद्वाज साहब जो हमारे गुरुजी हैं वो भी मौजूद हैं, सर के गुरुजी हैं वो भी मौजूद हैं। आप उनसे भी पूछ सकते हैं, आप तिवारी साहब से भी पूछ सकते हैं। तो प्लीज आप अपने आप को अनम्यूट कर लीजिए, और अनम्यूट करके जो क्वेश्चन है डायरेक्टली आप डॉक्टर साहब से बात कर सकते हैं और तिवारी साहब से भी बात कर सकते हैं। चैट बॉक्स आपकी बधाइयों से भर रहा है। भारद्वाज सर, जन्मदिन की बहुत बहुत शुभकामनाएं।
थैंक यू सर। थोड़ा सा अर्जेंट मीटिंग था इसलिए जाना पड़ा, बट एज आई सेड आई विल जॉइन अप whenever you give me time with an exciting topic on artificial intelligence in plant breeding and genomic selection.
जी सर, मैं आपसे कंसेंट ले लेता हूँ इस टॉक के बाद, जो भी आपका फ्री डे होगा उस पे मैं आपका कंसेंट ले लूंगा सर।
यदि है तो नो मीन्स इट्स ओके।
नहीं नहीं सर, डेफिनेटली सर, आपकी प्रेजेंस तो हमारे लिए बहुत बड़ा इंपॉर्टेंस रखती है। तो जो पार्टिसिपेंट्स पूछना चाहते हैं 1-to-1 क्वेश्चन इस पर्टिकुलर टॉपिक से रिलेटेड, तो डॉक्टर साहब आ गए हैं, आप प्लीज 1-to-1 करके अपने आप को अनम्यूट करके अपनी बात रख सकते हैं।
गुड इवनिंग सर।
गुड इवनिंग, वी बोथ आर देयर, प्लीज अद्वितीय।
यस सर। सर एक्चुअली ये बात कही जाती है कि पुराने टाइम में नॉर्थ इंडिया में काबुली चना और देसी चना काफी कल्टीवेशन हुआ करती थी। अब इसका डिक्लाइन क्यों है? ऐसा क्या रीजन है कि लोग अब सिर्फ वीट और राइस ही ज्यादा उगाते हैं और चिकपी और देसी चना पे इतना ज्यादा रिलाय नहीं करते?
देर आर इसके लिए काफी मल्टीट्यूड ऑफ रीजंस है, कोई एक रीजन नहीं है। सबसे पहले ये था कि हीलियोथिस जबरदस्त आती थी नॉर्थ इंडिया में, और उसका कोई ब्रीडिंग उपाय नहीं था। द ओनली सॉल्यूशन वी हैड वाज ट्रांसजेनिक, सो स्लोली इट शिफ्टेड इट्स फोकस। सेकंड ये हुआ कि अभी उसमें आईपीएम की काफी स्ट्रैटेजीज आ गए जिसके द्वारा आप कंट्रोल कर सकते हो, बट वंस इट मूव्ड अवे, एंड देन सेकंड मोस्ट इंपॉर्टेंट थिंग वाज इरिगेशन हैज कम इन ए बिग वे जी, है ना जी। तो इरिगेशन जब बिग वे में आया है और राइस ग्रो करने लगे हैं, और राइस के लिए एक एमएसपी दलहनी फसलों में, तिलहनी फसलों में भी है, पर एमएसपी पे शूर खरीदारी गवर्नमेंट करती है जो इन फसलों में नहीं है। तो इसलिए ये नॉर्थ से साउथ की तरफ आए हैं। और साउथ में क्या था कि यदि आप देखोगे कि खरीफ में रिमनरेटिव फसल थे, जैसे कि आपका सोरगम हो, या फिर मक्का हो, या फिर सोयाबीन हो। पर रबी में, वो भी असिंचित क्षेत्र में, सिंचित क्षेत्र की बात नहीं कर रहे, सिंचित क्षेत्र देखेंगे आप मध्य प्रदेश में, वहां भी धान होता है या धान गेहूं होता है। असिंचित क्षेत्र में जहां कुछ भी फसल नहीं होती थी, तो चना ने अपने आप को एडाप्ट कर लिया। साथ ही साथ यदि आप थोड़ा और नीचे जाएंगे, आंध्र प्रदेश में गवर्नमेंट ने एक्सलेंट इनिशिएटिव, और ब्रीडर्स ने भी एक एक्सलेंट इनिशिएटिव टुगेदर दिया। इनिशिएटिव क्या थी? एक जल्दी पकने वाली वैरायटी, आईसीसीवी 10 हो, है ना जी। तो एक जल्दी पकने वाली वैरायटी 108 से 110 दिन की विंडो इनको चाहिए था, तो उसमें जल्दी पकने वाली वैरायटी। फिर सिंचाई, एक सिंचाई, है ना जी। और तीसरा मोस्ट इंपॉर्टेंट है, जो एक खरगोन के एरिया में आप देखते हो रिवॉल्यूशन की तरह आया है, बोल्ड चिकपी काबुली, जो ये लोग डॉलर कहते हैं। अभी हमने भी रिलीज किया पूसा 40-35, भंडारण का स्रोत। आंध्रा में गवर्नमेंट ने किया, मध्य प्रदेश ने भी गवर्नमेंट ने किया, साथ ही साथ कुछ प्रगतिशील किसान किया। तो ये तीन ने चिकपी का रिवॉल्यूशन लाया है ना जी। तो इसलिए स्थानांतरण हुआ है। पर अब प्रॉब्लम हमें क्या आ रही है कि तराई क्षेत्र में हम चना को पुनर्स्थापन कैसे करें। तो इसमें मेजर प्रॉब्लम इज दिस कि नाउ बिकॉज ओवर इयर्स दीज आर हाई फर्टाइल सॉइल्स, और एक काफी हिस्सा में सैलिनिटी भी इफेक्ट होने लगी है। तो नाउ वी नीड टू डेवलप लाइन्स, हम उन प्रजातियों को या उन कल्टीवार्स को डेवलप करना है जो इनपुट मैनेजमेंट पे, हाई फर्टिलाइजर देने पे भी वो फर्टिलाइजर रेस्पॉन्सिव हो। चना तो फर्टिलाइजर रेस्पॉन्सिव है, पर बात है कि पल्ली छोड़ के पत्ते डालने लगती, यानी इनडिटर्मिनेट बन जाती है, और इसलिए फलियां या यील्ड कम हो जाती है। तो डॉक्टर नीरज इसी पे कर रहे हैं, ही इज वर्किंग ऑन सम डिटर्मिनेट लाइंस, जो हम हाई इनपुट कंडीशन में अभी हम लोग जस्ट डेवलप किए हैं। अब वो इनको हाई इनपुट का अगले साल से देंगे कि हाई इनपुट में भी ये ज्यादा ना बढ़े और पल्ली रखें। तो इस लाइन पे अभी रिसर्च कर रहे हैं, तो शायद कुछ दिनों में वी कैन ब्रेक दिस राइस स्वीट साइकिल सर। दूसरी बात है, आप राइस स्वीट साइकिल को भी ब्रेक कर लो, कुछ भी कर लो, आप किसान तो कोई व्हाट यू से इज नॉट अ फूल। he will take the variety or the crop which gives him more remuneration. सोयाबीन मध्य प्रदेश में क्यों था? क्योंकि उसको बहुत ज्यादा रिम्यूनरेशन मिल रही है। अब वीट के मुकाबले जिस दिन सिंचित क्षेत्र में चना में ऐसी यील्ड आएगी जो वील्ड को टक्कर दे पाएगी, तब जाके वी कैन रिप्लेस दिस। सो दीज आर टू चैलेंजेस फॉर द ब्रीडर सर, वी आर वर्किंग ऑन इट।
Okay,
बहुत
thank you.
बढ़िया सर, बहुत अच्छे से आपने पॉइंट रखा सर, बहुत अच्छे से चीजों को एकदम डेप्थ में जाके रिप्लाई किया। नेक्स्ट पार्टिसिपेंट यदि कोई
हेलो
पूछना
सर।
चाहे।
हेलो
किंग
सर।
बिहारी, हाँ जी।
सर्वप्रथम आपको जन्मदिन की हार्दिक शुभकामनाएं।
बहुत बहुत धन्यवाद जी।
तत्पश्चात हमारा क्वेश्चन है सर, क्यूटीएल हॉटस्पॉट रीजन क्या होता है सर?
नहीं नहीं, ऐसे देखें कि क्योंकि क्यूटीएल यानी, देखिए जीन्स तो आप समझते हैं आनुवांशिक
सर।
तत्व है जो कोई एक कैरेक्टर को कंट्रोल करती है ना जी। ए जीन इज
यस
अ
सर।
particular region in the chromosome which controls a particular trait or a group of traits through its expression through protein process, है ना जी। पर QTL जो है
यस
वो
सर।
स्मॉल इफेक्ट्स देते हैं, है ना जी। बड़ी जीन की तरह
यस
उतना
सर।
इफेक्ट नहीं देती है, छोटे छोटे इफेक्ट्स देते हैं और एडिटिव होते हैं। जब कुछ 4 या 5 एक ही कैरेक्टर बट हो सकता है कि जीन्स अलग अलग हो, एक ही जगह पे एकत्रित हो, दे आर प्रेजेंट एट ए सिंगल लोसाई, different group of genes that control a particular trait and are tightly linked, and they are actually passing from generation to generation without being disturbed, it becomes a QTL hotspot. अब जैसे कि आप नीरज जी ने, हमारे चने में भी है, धान में भी है। धान में है फॉर एग्जांपल द RH-T हॉटस्पॉट जीन, वो रिड्यूस्ड हाइट ही वीट में नहीं करती है, गेहूं में वो नंबर ऑफ टिलर्स बढ़ाती है, नंबर ऑफ ग्रेन्स बढ़ाती है ना जी। इसी तरह ये क्यूटीएल हॉटस्पॉट जो लिंकेज ग्रुप 4 या आजकल क्रोमोसोम 4 में है, तो वो क्या करती है, जो रूट की डेप्थ है वो बढ़ाती है, रूट की जो वॉल्यूम है वो बढ़ाती है। इन्होंने वो दिखाया भी है आपको, पूसा 362 में कम रूट्स हैं और ICC 4958 में डेप्थ एंड वॉल्यूम ज्यादा है। और इनकी जो हमने जब इसको डाला है पूसा 362 के बैकग्राउंड में, तो रूट की डेप्थ और वॉल्यूम बढ़ी। साथ ही साथ इसमें दाने की साइज का है एक है, साथ ही साथ हायर हार्वेस्ट इंडेक्स का भी जीन है, साथ ही साथ RWC और MSI कीप इट हायर अंडर वाटर स्ट्रेस, यानी इनसे ये सब वजह से इनको क्या होती है, जो लाइन इस जीन को रखती है वो शुष्क सहिष्णु यानी ड्राउट टॉलरेंट हो जाती है। अब ड्राउट टॉलरेंट उस क्यूटीएल को लाते हुए, इसलिए इसे हॉटस्पॉट कहते हैं, है ना जी। ऐसा ही विल्ट का भी एक हॉटस्पॉट है LG2 में। ये डॉक्टर तेरेसा मिलन हमारे कोलैबोरेटर है स्पेन से। यहाँ क्या है कि यदि आप जनरली विल्ट की जो रेसेस देखते हैं, तो 6 रेस इंडिया में मुख्य तरह पाई जाती है, वर्ल्ड में तो 8 है और भी ज्यादा है। तो FOC1, FOC2, फ्यूजेरियम ऑक्सीस्पोरम साइसरे, 3, 4, 5, 6 मेन रेसेस है। तो इसमें रेस 2 का जीन छोड़ के बाकी सब एक ही जगह है, साथ ही साथ 2 एस्कोकाइटा ब्लाइट के हैं। तो यदि हम ऐसी क्यूटीएल को विदाउट द बैकग्राउंड नॉइज ट्रांसफर कर सकते हैं तो हमारे लिए ग्रेट है। तो ऐसी जीनों की या क्यूटीएलों की एक जगह रहना और जनरेशन आफ्टर जनरेशन विदाउट कंटामिनेशन पास होने से, इनको हम एक लिंकेज ग्रुप में बांधते हैं और उस जगह को हम QTL हॉटस्पॉट कहते हैं।
बहुत
ओके
बढ़िया
सर।
सर। नेक्स्ट पार्टिसिपेंट प्लीज। मुझे लगता है कि लेक्चर ही अपने आप में इतना
कुछ क्वेश्चंस है, लेट मी लुक इन दैट। पाउडरी मिल्ड्यू इन चिकपी, आई थिंक दिस इज नॉट एन इम्पोर्टेंट वन। कहीं पाउडरी मिल्ड्यू रिपोर्टेड नहीं है चिकपी में? Now, I would like to know if there is any powdery mildew report in chickpea. This is first time I am listening. latest thing that is coming, which is spore developed, is rust, है ना जी. रस्ट के लिए कोई रेजिस्टेंट वैरायटी डेवलप नहीं हुई है. मेरे कोलीग डॉक्टर BS पाटिल जो हैदराबाद में स्थित है, वो और इक्रिसैट और हम लोग भी इन्वॉल्व है. वी आर ट्राइंग टू आइडेंटिफाई कौन सी टॉलरेंट सोर्स है. मेजर प्रॉब्लम हमें इसमें ये आ रही है कि रस्ट की रेसेस अभी वेल डिफाइंड नहीं है. हम अपने पैथोलॉजिस्ट फ्रेंड्स के तरफ देख रहे हैं, क्योंकि कहीं एक तरफ ये रेजिस्टेंट पाई जाती है एक जगह पे, तो दूसरी जगह ससेप्टेबल है. दूसरा है, अभी इसमें हमें पता नहीं कि वेदर ये 2 या 3 जीन से कंट्रोल्ड है, जैसे कि सोयाबीन रस्ट है या फिर वीट रस्ट है, कि या QTL कंट्रोल्ड है. उसका एक प्रोजेक्ट अभी पाटिल साहब को मिला है. सो सर्टेनली विद डॉक्टर ममता पांडे, डॉक्टर BS पाटिल, वी आर वर्किंग ऑन इट. दूसरा एक क्वेश्चन है, रेजिस्टेंट जीन कंट्रोल नहीं, पाउडरी मिल्ड्यू नहीं है जी, प्लीज, इट इज नॉट देयर. एक्सलेंट. एस्कोकाइटा ब्लाइट चिकपी में कैसे कंट्रोल करेंगे? एस्कोकाइटा ब्लाइट जैसे कि बताया, बहुत से रेसेस है, पर हमारे यहाँ 6 और 8 रेसेस है. हम लोग और पंजाब एग्रीकल्चरल यूनिवर्सिटी में पहले डॉक्टर सर्वजीत सिंह थे, अब डॉक्टर शैला बिंद्रा और इंद्रजीत सिंह के साथ हमारे पास एक कंबाइंड प्रोजेक्ट्स हैं, जिसपे हमने ऑलरेडी 2-3 वैरायटीज को हमने डिप्लॉय किया है. एस्कोकाइटा ब्लाइट क्या है, जनरली क्लाइमेट यदि कंड्यूसिव है तो वो पॉडिंग के समय आती है तो जनरली एस्केप हो जाती है, पर कभी कभी ये जस्ट पॉडिंग इनिशिएशन पे आ जाती तो जबरदस्त लॉस करती है. पर्टिकुलरली यदि आप देखेंगे तो धौला कुआं हिमाचल प्रदेश में और पंजाब में, जहाँ टेम्परेचर्स थोड़े कूल ज्यादा हो जाते हैं पॉडिंग के समय में, तो हमने इसके QTL जीन्स जो है, रेस स्पेसिफिक या QTLs का हो, आइडेंटिफाई किया है. पूसा 3022 है, पंजाब के भी 3-4 वेराइटीज है, तो इसमें हमने इन जीन्स को स्थानांतरित करके रिलीज किया है ना जी. तो वी हैव डन अ वेरी गुड वर्क. इक्रिसैट अगेन डॉक्टर राकेश श्रीवास्तव है, वो भी इन्वॉल्व है. अभी DBT ने नॉर्थ ईस्ट के लिए बॉटराइटिस ग्रे मोल्ड विथ विल्ट एंड एस्कोकाइटा प्रोजेक्ट भी दिया है. तो एस्कोकाइटा ब्लाइट 1 थ्रू मैनेजमेंट, देन नंबर 2 by deploying tolerant और resistant varieties, अभी काफी है, उनकी डिप्लॉयमेंट के थ्रू we can actually combat it. How can we control the viruses in chickpea? Now virus जो है चिकपी में मेजर प्रॉब्लम नहीं है. एक आती है वायरस, उसे कहते हैं ग्राजी स्टंट वायरस, हार्डली 0.1% इसमें रिपोर्टेड है, वो भी व्हेन टेम्परेचर्स आर वेट एंड कोल्ड. फ्लवरिंग के आगे, एक बार फ्लवरिंग सेट हो गई तो कुछ नहीं होती है. फ्रैंकली पुटिंग, वी हैवंट वर्कड ऑन इट. और एक क्वेश्चन है आपके यहाँ, एसिड टॉलरेंट जीन. नो, जब एसिड वेरी हाई रहता है तो इस जगह पे एल्यूमिनियम की टॉक्सिसिटी बढ़ जाती है. तो वी हैव जस्ट इनिशिएटेड वन प्रोजेक्ट. एक हमारे यहाँ डॉक्टर मिस्टर उमाशंकर है, हु इज वर्किंग ऑन दिस प्रोग्राम, एंड हम लोग नॉर्थ ईस्ट के साथ भी वी आर वर्किंग ऑन इट. अभी वी आर लुकिंग, कुछ वर्क डॉक्टर योगेश भाई, जो योगेश तिवारी है IAPR कानपुर में, और PAU में डॉक्टर शैला ने अभी थोड़े जीन्स और वेराइटीज या लाइंस विच आर टॉलरेंट टू हायर एसिड और लेट अस पुट एल्यूमिनियम टॉक्सिसिटी आइडेंटिफाई किया है. एक तो सॉइल एमेलियोरेशन प्रैक्टिसेस, दूसरा डेवलपमेंट ऑफ टॉलरेंट वेराइटीज. वी आर ऑन इट, नथिंग इज येट डन ऑन इट. एनीथिंग मोर सर?
सर सारे क्वेश्चंस का रिप्लाई बड़ी अच्छी तरीके से आपने कर दिया है, और अब तो कोई क्वेश्चन नहीं बचता, क्योंकि तिवारी साहब ने भी बड़ा अच्छे से एक एक चीज को रखा था, और बड़ा सेल्फ एक्सप्लेनेटरी वे में एक लेक्चर उन्होंने प्रेजेंट किया था. और जो चीजें बची थी उनमें आपने एक तरीके से जो पूर्णाहुति करके हम स्वाहा करते हैं, वो आपने पूरा काम कर दिया. तो सर बहुत बहुत धन्यवाद कि सर इतनी व्यस्तता के बावजूद भी आपने तिवारी साहब को जिम्मेदारी दी और फिर आके आपने अपनी बागडोर को संभाल लिया. ये वाकई काबिले तारीफ था सर. तो बहुत बहुत धन्यवाद आपके इस आशीर्वाद और आपके समय के लिए। और आगे जो आपने आर्टिफिशियल इंटेलिजेंस पे लेक्चर की बात कही है, उसमें भी सर हम जो है आपको डेट्स भेजेंगे, जो आपका शेड्यूल फ्री हो उस पे आप मुझे कंसेंट दीजिएगा।
जरूर
तो
जरूर। नमस्कार। हम
जी जी, बहुत बहुत धन्यवाद सर आपको। सभी आपको पुनः जन्मदिन की शुभकामनाएं, और तिवारी साहब को पुनः बधाई यंग साइंटिस्ट की।
थैंक
सभी
यू।
को जय श्री राम, बहुत बहुत धन्यवाद। सभी पार्टिसिपेंट्स लेफ्ट
जय
कर
श्री
सकते
राम, जय श्री राम, नमस्कार। वी आर लिविंग सर।
चलिए।